Okay, hello. Today we'll start our second lecture. You can interrupt me anytime for questions, okay? So last lecture, we concluded the lecture by the classification of toxic agents. And we understood that there are two major definitions, the toxin and the toxicant. And we understood the difference between both these definitions, and we gave some examples. As well, we knew that some compounds could be named a toxin and a toxicant as well, based on its source. Okay. Today we'll start go deeper into the course of the mammalian toxicology, and we will go to understand some what are the spectrum of the undesired effects that caused by the toxins or toxicant. So we'll start with the definition. What is allergic reaction? So basically, allergic reactions, it's all about the immunology, the immunity of your body. So it is immunologically, so that's why it's mediated by the immune system. And due to the exposure of these toxic agents, it can generate adverse effect to any chemical that is foreign to the body. And this can be as a result of initial exposure to that chemical or previous exposure. And of course, the intensity of the action of the immune system will depend on the concentration of the chemical and how many times this chemical will expose to the organism. So usually the expression hypersensitivity is most often used to describe this allergic state. So what about idiosyncratic reactions? Actually, this one is different because basically it depends on the genetic background. So it's a genetically determined abnormal reactivity to a chemical. And the response can be observed qualitatively similar to that observed in all individuals. Because since it is based on the genetic background, so if there is a defective gene in one individual, the same defective gene in another, the same gene is defective in another individual, the response could be similar. The response to that chemical, which is affected by the defective gene, will be qualitatively similar in these two individuals, for example. Then we need to differentiate between two expressions, the immediate versus the delayed toxicity. So we have the immediate toxic effect. And of course, from the name implies that these effects develop very rapid after only single administration of a substance, while the delayed toxic effect affect those that occur after some time. فاهمين الفرق يا شباب بين immediate والdelayed ولا لا؟
Yes, doctor.
Yes, doctor.
اوكي، حد يقدر يدي مثال كده من خبرتنا من معلوماتنا العامة كده؟
immediate يمكن شيء إنه مثل البنج أو هيك. delayed مثلا مثل البيلز أو لا
الايه؟
البيلز مثلا الأدوية وهيك.
لا، خلينا نقول مثلا واحد ممكن ياكل انتوا عارفين ان في بعض الناس ممكن يجي لها حساسية من الشوكولاتة، ممكن ناس يجي لها حساسية من الفراولة، صح كده ولا لا؟
Yes.
ممكن واحد ياكل حاجة يظهر ألرجي على الجلد بتاعه على طول، صح كده ولا لا؟
Yes، مثل طفر جلدي أو شيء.
نعم؟
مثل طفر جلدي.
افتكرتي ايه؟
طفر عم أقول.
فهمت. في واحد ممكن يتأخر الريسبونس، صح كده ولا لا؟
Yes, doctor.
اوكي؟ اوكي نكمل. So what about the reversible versus the irreversible toxic effect? So basically, it is, you can understand from the name by itself. So if a chemical produces a pathological injury to a tissue, if the ability of that tissue to regenerate, it will determine if this toxic effect is reversible or irreversible. One of the very important organs in our body is the liver. And liver actually is equipped because it is equipped and can regenerate itself because actually it can metabolize a lot of toxins inside our body. So as I just said, liver has a high ability to regenerate, because that's why any injuries in the liver are reversible, while central nervous system, any injury in that system, it will be largely irreversible, because of course, as you know, the central nervous system, the cells cannot be divided. لمعلومات حضراتكم برضه لازم تبقى عارف ان جسمنا فيه عدد نيرف سيلز محدد لا يتغير من ساعه ما حضرتك بتتولد. عارفين المعلومه دي ولا لا؟
يس اكيد.
اكيد. فالنيرفز اللي هي بتكون السنترال نيرفس سيستم عموما، اي نيرف موجود في جسمنا انت مولود بيه، وبيحصل الترمينيشن للسيل سايكل at some point during the development, so we have a fixed number of nerve cells. So any damage in these nerve cells, it will not be restored or regenerated. Okay. And these two examples differentiate between the term reversible and the irreversible toxic effect. So carcinogenic and teratogenic effects of chemicals, these actually which affect the organism and lead to formation of cancer, are usually considered irreversible toxic effect. So what is the difference between local versus systemic toxicity? As well, these are familiar definition. Of course, the local, it will affect at the site of the first contact between the biological system and the toxicant. While the systemic, from the name, it affects larger area, so it requires the absorption and distribution through all the body of the organism, and it will the toxicant, once absorbed and distributed all over the body, so it will exert its effect at a region that is very far, relatively very far from the first site of exposure, and could have very harmful effect. So let's talk now about the characteristics of the exposure to toxicant. So to understand that, we need to understand the route and the site of exposure. So the major routes by which toxic agents gain access to the body, they are all known, either by ingestion through the GIT, or by inhalation through the lungs, or topical or dermal through the skin, or parenteral other than intestinal canal routes. So these toxic agents, in general produce the greatest effect and the most rapid response if they are given directly to the bloodstream. اوكي؟ ودي نقطة احنا كلنا عارفينها. لما واحد بياخد مسكن، صح؟ ايه اشهر المسكنات يا شباب؟ حد عارف اسامي المسكنات؟
بنادول،
الباراسيتامول، ايه تاني؟
سرقام، اشياء للتوث ايك.
تمام. ايه تالت؟ الكيتوفان، صح كده؟ او الكيتولاك،
البروفينول يعني،
او البروفينولز، الكيتولاكس كلها. الكيتولاك ده مش ممكن تاخده تابلت؟
بلى
يس،
بس باسم تاني طبعا. وممكن تاخده انجكشن في الدم، صح؟
اكيد بالمصل او شيء.
لا ماسلز هيبقى اسمه انترا ماسكولار.
لا مش
لكن
مصل.
لو خدته بالدم، طبعا في البيت لو حد لا قدر الله بياخد انجكشن لاي مواد من عائله الكيتولاكس دي، بياخدها ماسلز. فاهمين قصدي؟ لكن لا قدر الله لو واحد راح المستشفى بيحطوا له في المحلول اللي هو بيخش انترافينوس، صح كده ولا لا؟
يس
صح.
تمام. طب يبقى لازم نبقى فاهمين ايه؟ ان الافكت لما يبقى انترافينوس هو افكت سريع جدا عن ان انا اخد التابلت اللي هي بتاعت الكيتولاك. فهمتوا؟ اوكي.
اوكي دكتور.
So let's talk now about a descending order of the effectiveness for the routes. يبقى احنا هنقارن الراوتس كلها ونعرف مين اللي اسرع حاجه، وبعد كده بتقل العمليه. خلاص؟ هتلاحظ هنا ان عندنا الانهيليشن هو اسرع حاجه، intraperitoneal, subcutaneous, intramuscular, intradermal, oral, dermal. يعني هنلاقي ان الاورال بيجي لو انا خدت دراج او خدت او توكسيكنت انا حصل لي بلعته بالاورال، الافكت بيكون او السيفيريتي اوف او الافكتفنس بتاعته بتقل كتير جدا عن ان انا عملت له انهيليشن. فاهمين الفرق ولا لا؟
Yes, doctor.
Okay. So there is something called the vehicle. What is the vehicle? Actually, the vehicle, it is the material or the liquid, basically the liquid in which this chemical is dissolved. And this have a very significant effect on the absorption efficiency of that toxin after ingestion or inhalation or topical exposure. كتير مننا يا جماعه كانوا بياخدوا كان بيجي حالات ياخدوا فيه انتبيوتيك، صح كده ولا لا؟ ساعات كنت بتاخد الانتبيوتيك كتابلت، يبقى كده اورال، كله اورال تابلتس. لكن في بعض الانتبيوتكس بتيجي على شكل باودر، وبتحتاج ان انت تحطها في ميه او ديستلد ووتر بتيجي مع الانتيبيوتيك. حد شاف النوع ده من الادويه ولا لا؟
بلا دكتور اكيد.
الميه او الستيرل ووتر اللي بستخدمها دي عشان ادوب بيها الباودر بتاع الانتيبيوتيك دوت، دي او الانتيبيوتيك دي ما هي الا الفيكل. فهمتوا مصطلح الفيكل هو ايه؟
Yes.
Okay. So the route of administration can influence the toxicity of the agent. Very clear sentence. ليه؟ لأن احنا لو عندنا نفس التوكسيك ماتيريال دي أنا عملت لها انهيليشن، السيفيريتي بتاعت التوكسيستي بتاع هذا الايجنت will be so much high than ان انا لو خدتها اورال. اوكي؟ عشان كده برضو يمكن اقول لازم اقول مثل مهم هنا ايه هو؟ ان الناس اللي بتتعاطى المخدرات يا شباب، فاهمين قصدي ولا لا؟ خلي بالكم احنا لما بنقول شباب قصدنا الشباب والصبايا، فاهمين؟ اوكي؟ اوكي. طب عشان كلكم صبايا هنقول صبايا بقى. خلاص؟ انتوا عارفين الناس اللي بتاخد المخدرات، مش في فتره مش في الناس دي ممكن تاخد المخدرات على شكل تابلتس؟
بلا.
Yes.
بيجي كلنا بنتفرج على الافلام وبنشوفها، بيجي في الافلام بقى بيجي مرحله ان التابلتس ده ما بيأثرش معاه، صح ولا لا؟ فبيروح جاي عامل ايه؟ يروح جاي طاحن التابلتس دي، عامل لها جرايندنج ويعمل لها ايه؟ انهيليشن. فهمت ولا لا؟
Yes.
تمام. يجي الانهيليشن بقى ما ياثرش معاه، يعمل ايه؟ لو انتوا فاكرين من الافلام اللي كنا بنشوفها يعمل ايه؟ يروح طاحن ايوه، يروح طاحن التابلتس دي ومدوبها في ميه وحقنها في الوريد، صح ولا لا؟
Yes.
Okay. So let's talk for an example how the route will affect the toxicity of an agent. Let's talk an agent that acts on a central nervous system, okay, but is efficiently detoxified in the liver. يعني هذا الايجنت او التوكسيكنت ده لو وصل الليفر هتقدر تشيله من الدم، بالتالي مش هيبقى له تاثير، صح كده ولا لا؟ لكن لو وصل للسنترال نيرفس سيستم هيشتغل عليه جامد. خلاص؟ هنلاقي ان هذا الايجنت which act on a central nervous system is less toxic when given orally than when given by inhalation, via inhalation. وده كلام مفهوم جدا ليه؟ Because the oral route requires that a lot of the dose will pass through the liver before reaching the circulation and then the nervous system. وده لو انتوا حضراتكم عارفين الدايجستيف سيستم او البلاد سيركيوليشن، لازم تبقوا فاهمين ان منطقه الجي اي تي كلها بيطلع منها فينز بتلم الدم اللي طالع، الفينز طالع منه اللي هي بيحصل ابزوربشن طبعا في الانتستين، والانتستين الابزوربت ماتيريال اللي في الانتستين دي بتتلم فينز، وهذه الفينز بيحصل لها collection in the liver for the process of detoxification. Then the liver will pump this blood to the heart for the general circulation. يبقى we understand that in case of the route exposure, the toxicant have to pass the liver in order to be toxic, okay, and in order to reach the nervous system. So that's why it will be less toxic to the central nervous system in case it have been taken through the oral route. فهمتوا يا شباب النقطة دي ولا لأ؟
Yes, doctor.
Okay. So we have either direct contact, either by ingestion or either by inhalation. So there is something called occupational exposure to toxic agents, and these are mostly result from breathing contaminated air, inhalation. زي ان الواحد يبقى عايش في منطقه ملوثه بعوادم السيارات، او الناس اللي بتشتغل في المناجم الفحم مثلا، او المناجم اللي بتطلع اي مينرال والكلام ده كله. Because they can inhale the contaminated air through inhalation, or can be affected direct and prolonged contact of the skin with the substance. يعني ممكن يكونوا شغالين في الفحم، في مناجم الفحم، وهذا الفحم بيتراكم على الجلد بتاعه، ممكن يأفكت السكين ده through the dermal exposure. Whereas some accidental and suicidal poisoning occur most frequently by oral ingestion. معظم الناس اللي هي بتنتحر او accidentally ياكل سم، وكان في زمان لو تفتكروا حالات كتيره جدا ان لا قدر الله يا اما اطفال يا اما ستات يبلعوا سم فران بالغلط، صح ولا لا؟ Okay, and usually by oral ingestion. So if you compare between, and you need to understand the comparison of the lethal dose of a toxic substance by different route of exposure, this will often give a very useful information about the extent of absorption. So if you have the same compound and you need to check what is the lethal dose, you need to compare the effectiveness of this compound through a different routes of exposure. Of course, one of them is through the oral exposure, oral ingestion. So to understand how the difference between the lethal dose based on the route. So when the toxic dose after oral or dermal administration, if these toxic dose after the oral or dermal administration is similar to the toxic dose after intravenous administration, this means that this toxic agent is absorbed very rapidly. يعني بمنتهى البساطه لو انا عندي كومباوند حد خده اورال وحد خده انترفينس والافكتفنس تقريبا واحده او بيأثروا نفس الضرر الاثنين، ده معناه ان هذا التوكسيك كومباوند is absorbed very rapidly وكان bypass عمليه الليفر detoxification. فهمتوا يا شباب؟
Yes, Dr. Clear.
Okay. Conversely, وبطريقه عكسيه، لو التوكسيك دوز باي ديرمال راوت، يعني انا محتاج دوز كبيره جدا is much higher than the oral dose، معناها ايه بقى هنا؟ ان السكين can provide an effective barrier to the absorption of the agent، لان هذا الايجنت ما قدرش يخترق ايه؟ السكين. So let's talk about duration and the frequency of exposure. So toxicologists usually divide the exposure of experimental animal to chemicals into 4 categories. Okay, these 4 categories actually acute, subacute, subchronic, and chronic. The acute exposure, it's from the name, so it's a single exposure to a chemical for less than 24 hours. Okay, so if there are any repeated exposure, so you go to the other 3 definition, either subacute, subchronic, or chronic. Okay, then we have the subacute. Actually, it is as well repeated exposure to a chemical, but for 1 month or less. For the subchronic, the period can reach from 1 to 3 months, and for chronic exposure, it is extended over than 3 months. So these are the categories of the exposure and how they related to the period of time of exposure to the name: acute, subacute, subchronic, and chronic. So let's see an example. If the agent is ingested, it will pass through the mouse GIT tract, and a lot of it, if it's poorly absorbed, it will go outside from the feces. If there is inhalation route, it will go through the nose and mouth, lungs. It can go through from the lungs to the blood. If it passes to, it can go through from the lungs to the blood. That's why the inhalation have an effectiveness much more than the ingestion. If you go for the skin absorption, it can go to the fat, and then from the fat it can go to the sweat and mammary glands, and it can be secreted and detected as well. This agent or this toxicant can be detected in the sweat, in milk, in tears and saliva. Actually, these are the secretions of these glands. It can go directly as well to the blood and lymph. And from blood and lymph, it can take the same route of the fat route. And if it is transported to the blood, it can pass through the liver or through the kidney, and it can be excreted as well by urine. Of course, the process of excretion or got released in the secretions or in feces depend on a lot of factors. One of the major factors is the bioavailability, how much it will stay in the blood, what is the efficiency of the absorption of this compound through the GIT tract. So let's talk here about some explanation about, in this graph, we'll see the relationship between the dose and the concentration at target site under different condition of dose frequency and elimination rate. So we'll understand now, at the x-axis we have time, increasing time, and at the y-axis we have concentration at the target site. So that's mean that the toxicant or the agent have what is the concentration which is measured at the target site. So for example, if you have a toxicant that will reach liver at some point. So in this graph, they measured the concentration of that drug in the liver, okay? And that is the target site. Now we have three chemicals, A, B, and C. These are three actually different chemicals or different toxicants, or you can assume it's different drugs. To differentiate between these three chemicals, chemical A, it have a very slow elimination. It is a resistant chemical. So it is removed very slowly from the circulation. While chemical B, this chemical, its rate of elimination equal to the frequency of dosing. So because you increase the dose by time, and since there is equal relationship, so for some reason it will keep a constant level. For C, this compound, its rate of elimination is very fast than the dosing frequency. So these three different compounds, you can understand these facts actually from the graph, because over time, at this arrow, let's have a pointer. At this time point, represented by the arrow, the dose was administered to the organism. Three compounds, A and B and C. Over time, you can see over time that A is very stable. That's why it has a slow elimination rate. While C dropped in concentration. That means its rate of elimination is much faster than the dosing frequency. While B, for some reason, it says in the middle, because there is equal relationship between the rate of elimination and frequency of dosing. Okay, so the blue shaded area here, the blue, represents the concentration at which the chemical at the target site needed to have a toxic effect. Okay, so if you look here, so this blue shaded blue shaded box represent the concentration that is needed to provoke a toxic response. So you can understand from this figure that A remains a lot of time, so that's why it will provide a lot of toxic effect. While C almost disappeared from the range of concentration that causes the toxic effect, B have an average effect on the target site. فاهمين يا جماعة؟ قبل بس ما أكمل الكيرف، فاهمين قصة الكيرف ده ولا في حد عنده سؤال فيه؟
No, doctor, all clear.
Okay, let's do some experiments here. Okay, let's see now it's happening. If you understand here, this figure represents a single dose. So that's the effect, or that's the concentration over time after administration of a single dose. What happens if we try to administrate repeated doses? Repeated doses for the same 3 drugs. So you can see here the arrows represented the time point where the doses was administered. شباب قبل ما اشرح بس الفيجر ده خلينا بس يعني عشان اقرب لكم الفكره، خلينا بس نقول حاجه. هو مش فينا بعض الادويه الدكتور بيقول لك خدها في اليوم مره واحده، صح؟
صح.
دي بتبقى سنجل دوز. طيب وفي بعض الادويه الاخرى الدكتور بيقول لك ايه؟ خدها 3 مرات في اليوم، صح؟ طيب ليه تفتكر ليه الدواء ده قال لك 3 مرات في اليوم وليه ده قال لك مره واحده؟
حسب الدوز قد ايه كان.
لا يعني ماشي، ما هو يعني نعم؟ لا الصوت بيقطع.
كرمال الافكت تكون اعلى تبعه.
خلينا نقول بقى بطريقه احسن، الدوز اللي عطاه لك، خلينا نقول عشان نقرب الامور لنفسيكم، الدوز اللي عطاها لك مره واحده في اليوم ده معناها ان الدراج ده الابزوربشن ريت بتاعه عالي جدا. خلاص؟ لكن الدراج اللي عطاه لك 3 مرات في اليوم، ودي كرايتيريا الايفكت بتاعه.
انه حسب الايفكت، اذا ايفكت اول ناخده مره.
يبقى بالتالي الايفكت ده انا محتاج اديله 3 مرات. ليه؟ لان انا محتاج اعلي الكونسنتريشن بتاع هذا الدراج عشان يبقى في الدم كافي ان يبين الايفكت بتاعه. فهمتوا؟
يس.
طيب واللي خلينا نفهم الكلام ده اكتر، بصوا للشيدد اللي هو البلو شيدد بوكس ده. علشان الدراج يبان له افكت المفروض الكونسنتريشن بتاع الدراج يبقى في منطقه الشيدد بوكس ده، صح كده ولا لا؟ طيب لو افترضنا ان الوقت هنا كان 10 ساعات، انا بتكلم في السنجل دوز 10 ساعات، ده معناها ان الافكت بتاع الدراج ايه؟ هيقعد في التارجت سايت قد ايه وقت؟ 10 ساعات، صح؟
10 ساعات صح.
طب بالنسبه لبي هيقعد قد ايه وقت؟
ممكن
5 ساعات، صح كده؟ طب بالنسبه لسي هيقعد قد ايه وقت؟ تقريبا ساعه. فهمتوا الفرق وعلاقه البوكس اللي مرسوم بالازرق ده عشان تبقوا فاهمين الافكتفنس بتاعت الدراج ولا لا؟
يس.
طيب تعالوا نشوف بقى التجربه. عملوا ريبيتد دوزز، نفس الجايد لاينز. ده دراج ايه، ده بي، ده سي. هنكتشف ايه؟ عشان الدراج ايه يبقى افكتيف انا محتاج كام دوز؟
5 دوزز.
ليه 5 دوزز؟
الدراج A دكتور 3 دوزز.
لان مع الدوز الثالثه الكونسنتريشن دخل جوه البلو بوكس، صح كده؟
يس.
طيب بالنسبه للدراج بي انا محتاج كام دوز؟
5 دوزز.
5 دوزز. طب بالنسبه لسي؟
ما لنا افكتيف ابدا.
يبقى for C a toxic concentration will never be reached regardless of how many doses are administered. صح ولا انا غلطان؟
صح.
تمام؟ فهمتوا هنا؟ خلي بالكم هنا احنا بنتكلم على توكسيكنت، لكن هو نفس المنطق بتاع الدراجز برضه. فهمتوا ولا لا؟ اوكي؟ في حد عنده اي سؤال في السلايد دي؟ اوكي؟ طيب يا شباب So now we'll talk about the dose response relationship, okay? So actually this dose response relationship will understand, or we correlate the relationship between the exposure, the characteristic of exposure, and the spectrum of the toxic effect. And actually there are two types of dose response relationship, okay? We have the individual dose response relationship. So basically, first thing to know, it is related to one individual, in which it is having an increase, as you can see from the x-axis, it have an increasing dose of something, drug, toxicant, whatever, and it will affect the response, which in these cases, for example, the drug is which is taken, it will affect the activity of enzyme cholinesterase or the carboxylesterase. It will affect its activity. So you can see once you increase the dose, the percent inhibition is increased. Okay, so that's why in the individual dose response relationship, you can see there are a graded response based on the increasing and the continuous administration of a range of doses. So basically, for convenient, we can transfer this line chart into straight line chart. This can be done by converting the X scales into log scales to the base 10. When you convert this scale into a log scale to the base 10, you will now have a best fit line, and you can see it is represented by a straight line. And actually, these two graphs represent two ways to represent data. And this is the individual dose response relationship. What about the other type? The other type is called quantal dose response relationship. The first one dealt with the one individual, but for the quantal dose response, it deals with a population. So it characterizes the distribution of the individual responses at different doses in a population of these individual organisms. So a widely used statistical approach for this relationship to estimate the toxic exposure is called the effective dose. And it is usually used for drugs to understand the effective dose that is needed to have the appropriate response. So usually the scientists decided to use the effective dose indicator 50%. So it's called ED50 value. So this ED50 value will represent 50% of these individuals have the response level, or 50% in this population got the perfect response level. So if you need to measure the death, which is caused by a certain toxicant, so in these cases, it's not called ED50, it's called LD50, which is the lethal dose 50 that is responsible for killing 50% of the individuals. So because this quantal dose response phenomena is based on individuals in a population, some of these individuals will show low response. Some of these individuals will show high response. And if you compare these responses, for example, with respect to doses, you can see a binomial distribution. And what's the mean of binomial distribution? It means you can see a histogram that is increasing, then decreasing. In other words, it can be represented by an inverted bell shape graph, a line chart, inverted bell-shaped line chart. You have some of the population which will show 20% response. Let's say, for instance, a population that is middle age individual in the population show 20%, while children, which is below puberty, it shows like around 10%, and over age will show as well 10%. So this will give us the clue that the perfect match for the appropriate response for this individual. Okay, so in order to be more, the approach to be more easier for the understanding, so, and because the distribution is normally distributed in the shape of inverted bell shape, so we can convert the percentage response to units, which these units can deviate from the normal mean of the population, which is M. So normally the mean for the 50% response, normally the mean will be 0, because we have negative values and you have positive values. Some of them will have negative value for the response or positive value for response. So we have a mean of 50%, which is 0. So normally, for convenient, we add 5 to the value of the mean to avoid to have negative numbers. So these converted units, now it's called profit units, because we use the mean, we add the 5 weights. So a 50% response become a profit of 5. What about if the mean increase? So it will add +1 deviation from the mean and become a profit of 6. What if the mean have a negative decreased by -1 deviation from the mean? So it will subtract from the 5 and it will be 4. This for convenient, and to draw a much better line chart, straight line best fit chart, you can see that on the x-axis the concentration, and you have 2 y-axis. One of them represents the profit values after being adjusted, and the other one actually represented the older version of the percent response. Okay? Any questions?
No, doctor.
Okay. So let's see here how the dose-response relationship for a substance that are required for normal physiological function and survival, like vitamins, for instance. So you can see the shape of the graded dose response relationship of an individual over entire dose range take the shape of a U shape. So actually this is actually increasing dose on the x-axis and the response on the y-axis. You can see that actually the curve, the inverted shape, it's only for one individual. So you can see when you increase the dose, you come at one point that the dose is very toxic. And the best response for that dose, the best response of that dose can be seen at the region which is the region of homeostasis, the blue region. This is the best response, because lower than that, the dose will be deficient to provoke any response. And above this region, it will be toxic and will produce over response, which will be harmful for the individual. You can see here, these are examples of a lot of chemical compounds, and these are the calculated LD50 or the lethal dose 50 for this compound. You can understand now that in order someone to die from the ethyl alcohol or the ethanol, it need to drink at least 10,000 milligrams per kilogram to die. Okay, so it's a huge amount, of course. For the sodium chloride, and you know the sodium chloride is the salt we use in our food, it needs almost 4 kilograms to be a lethal dose, and can be when consumed by individuals. For substance or chemicals like botulinum toxins or dioxins, only very trace amount will be very toxic. فهمتوا الفاريشن يا جماعه في ال دي 50 دي في الكومباوندز ولا لا؟
Yes, كتير.
بس مش مطلوب منكم تحفظوا الارقام ديت. اوكي. You don't need to memorize this number in that table. Okay.
Okay, doctor.
Okay. So let's see here. What are the assumptions in using those relationship, those response relationship? Actually, the response is due to the chemical administrated. This one of the assumption, of course, if you have a response, so it is because of the chemical that have been administrated. The magnitude of the response is related to the dose, of course. If the dose is high, then the magnitude will be high of the response. So actually, this magnitude of the response, it's a composite of three other assumptions. This compound or this chemical, once it is internalized inside the body, there is a molecular target site or sites that the chemical will interact to initiate that response. That's first assumption. Maybe the production of a response and the degree of the response are related to the concentration of the chemical at the target site. In other words, maybe you have a huge concentration of a chemical is administered to the organism, but only trace amount have reached the target site. So you will have decreased response. So these assumption is important in understanding. To have a response, you need to measure the concentration of the chemical at the target site. The third assumption that goes to the magnitude of the response is the concentration at the site is related to the dose administered. And as we said in the previous slide, it is different. The route of administration reflect how the concentration of that compound will be at the target site. Okay. The third assumption, there exists both a quantifiable method of measuring and the precise mean of expressing the toxicity. If we know and understand all these assumptions, we will never go into a conclusion until we know how to measure the response and how to measure how these toxic compound have led to the toxicity. If you can't measure the response that have been provoked by the toxic compound, you will not understand the dose-response relationship. For example, a chemical that produces cancer through genotoxic effect. What's meaning a chemical that produces a cancer through genotoxic effect? Some chemical can bind to DNA, causing DNA mutation, which may lead to cancer. This chemical as well can cause liver damage, can cause a central nervous system effects via different mechanisms. So it can cause a lot of effect into these three organs, or these three molecules or organ by different mechanisms. The same chemical which causes this toxic effect at different target sites may have three different dose-response relationship, one for each endpoint. في حد عنده سؤال يا شباب لغايه دلوقتي؟
No, doctor.
Okay, let's now evaluate the dose-response relationship. Let's take for example the anesthesia. Okay, عارفين الانستيزيا هي ايه؟
Yes, doctor.
Okay, so let's see here. We need to calculate very important index here. We need to compare between the line chart of each of these three different doses. We have the one for the effective dose, and one for the toxic dose, and one for the lethal dose. So you can see here in the line chart, with increasing doses, we are measuring the response as a profit unit. Okay. So the first curve here, let's have a pointer. So the first curve here represents the ED, the effective dose curve. While the second line chart here represents the toxic dose. While the third line here represented the lethal dose. And of course, it is corresponding increasing in concentration. Okay, so how this will help us to identify or calculate a very important index. It's called therapeutic index. Therapeutic index. For the TI, this therapeutic index for any drugs, which is actually its approximate statement about the safety or the relative safety of that drug, and should be expressed of a ratio of the toxic dose, 50% toxic dose, over to that of the effective dose, okay, over the effective. For example, from the figure, you can approximate the therapeutic dose. How is that? And you need to know the larger the ratio, the greater the relative safety. Okay, you can see from the curve, the ED50 can be calculated. How, of course, we have said that 50% represent 5 in the profit. Okay, if you remember from the last few slides, 50% represent 5 in the profit units. So the ED50 will be around 20, as you can see from the figure. While the TD50, the toxic dose, TD50 will be around 60. So how we can calculate the therapeutic index now? For the therapeutic index, it will be 60 over 20 based on this equation. So it will be 3. So if we're comparing two drugs, both of them have the same effect, have the same therapeutic effect. We need to calculate the therapeutic index for both of these drugs. If one of them came up with 3 and the other one came up with 5, which of these two drugs will be safer? حد يقدر يجاوب؟ حد يقدر يجاوب which of these drug will be safer? شباب
اللي عنده higher يعني ال5.
تمام، يبقى اللي عنده 5 الثيرابيوتيك اندكس بتاعه 5 هيبقى سيفر عن اللي عنده 3. اوكي، based on the equation. عرفتوا ازاي تحسبوا يا شباب الموضوع دوت؟
يس دكتور.
عشان لازم بسأل سؤال منها في الامتحان. اوكي؟
اوكي.
So now we'll move to the last topic of our lecture today, which is called toxogenomics. Toxogenomics. So in other words, how the toxic compounds will affect the genetic background and how this is assessed to understand the response based on the response on the DNA. So in past decade, numerous new genome-based technology have become available. These technologies allow large-scale analysis of biological response to external stimuli. يعني معناه ايه الكلام دوت؟ ان خلاص في السنين الاخرانيه بداوا العلماء مش هياخدوا دراج واحد ويشوفوا الريسبونس بتاعه، هياخدوا دراج واحد وعايزين يشوفوا الريسبونس على الجينوم او على الدي ان اي. العمليه هتبقى consuming, time consuming وmoney wise consuming as well. So you don't need to take one drug ويشوفوا الريسبونس على الدي ان اي، هو طبعا بيشوفوهم على الانيمالز الاول. خلاص؟ فبالتالي لا اصبح انا اقدر اعمل large scale analysis. ازاي؟ زي ما هنشوف لاكتر من دراج عشان اقدر اعرف الايفكت بتاعه على population. So the traditional scientific approach عشان اقدر اعرف ال biochemical and molecular effect of these toxic substances focused on examining the biochemical pathway. أي biochemical pathway connected to the observed response can be identified either by pathology, histology, blood chemistry, or even a behavioral observation. ودي الطرق القديمة إن كانوا يحطوا الدراج ويبداوا يشوفوا الريسبونس او كيميكال، ويبداوا يشوفوا الريسبونس على المستويات دي، على البايوكيميكال باثويز والموليكولار ايفنتس either by التراديشنال ميثودز زي الباثولوجي والهيستولوجي والبلاد كيمستري، يقيسوا يعملوا تحليل دم، يشوفوا بيهيفير اتغير نتيجه للدراج ولا لا. عايزين يشوفوا الافكت بتاع الكومباوند. However, these new technologies which are available now can examine the entire or the universe of biological response to a toxic substance. اوكي، اقدر ادرس هذه التكنولوجيز بقى الجديده اللي هنتكلم عليها، تقدر تطلع لي كل البيولوجيكال ريسبونسز اللي حصل. طب ازاي؟ التكنولوجيز دي انكلود ايه؟ الجينومكس دي ازاي بتعمل كاركترايزيشن للجينوم، الترانسكريبتومز ده ازاي بتعمل كاركترايزيشن لاول messenger RNA expressed in cell tissue in just one shot. Proteomes as well for proteins. Metabomics, we characterize the small molecules, the metabolites that the byproduct, the substrate, the cofactor and enzyme reaction that could be developed as a result of a toxin. There is a lot of omics here. You have lipidomics, you have nutrigenomics, and this actually will deal with the effect of nutrients on the DNA. Because each level of analysis generate a very large quantity of data, so you need another branch of science which is called bioinformatics. And in bioinformatics, you combine all data that is collected. You do organization, you do evaluation, you do statistical analysis. All of these approaches is called collectively bioinformatics. And this bioinformatics branch of science has been developed to address this lot of data, this whole many computational and statistical challenges. So in the field of toxicology, of course, there is toxicogenomics. And this term is used to define the area of research only that combine all the info coming from the transcripts, messenger RNAs, proteins, metabolites, conventional toxicology, all of them combined together, investigate the interaction between genes. All of these will lead to a single information that reflect a genome-wide analysis. So to incorporating this new technology into toxicology, you need to understand several keys of components in order to get to come into the information that is needed. So you need a large database, very large database of treatment-specific information, such as analysis from target tissues, body fluids derived from toxicant-treated animals. You need as well genomic database that describe DNA sequence from species of interest. You need computational tools that extract the information from database and as well from the published literature to identify critical pathways and networks that affect or are altered by the toxicant treatments. And finally, you need to compare the traditional toxicological endpoints that came from histology, pathology, blood chemical tests to ensure that the observed omics responses are closely aligned with the toxicant-related pathophysiology, of course, in the animal. And this process is called phenotypic anchoring. في حد عنده سؤال يا شباب لغايه دلوقتي؟ فاهمين الاربعه ابروتش دول عشان اقدر اعرف اطلع بنتيجه ادخلها تبع التوكسيكوجينومكس؟ في حد مش فاهم الاربعه دول؟
دكتور اوكي ثانك يو.
Okay. So this pattern, if you have all these data and you saw a pattern of a change in the transcript, protein, metabolite profiling, this will provide a signature for the toxic response. Okay. So now we are talking about if a toxic agents, and we have all this info that came from different sources, and we analyze and interpret, and we collect the information from the literature or published studies, and from animal studies, and from human studies, and we analyzed all these inputs for this specific toxin or drug, so we will have a signature for that drug. of this toxic response that will be great value in predictive toxicology. So now I can predict something that will occur for a compound, and it is very useful in pharmaceutical development. So the introduction of omics technologies to toxicity, actually, to toxicity testing will contribute to very important thing. Will contribute to the reduction, to the refinement, to the replacement, and actually it's called the 3 R's of animal in toxicity testing. Okay, so it generally هتقلل عدد الحيوانات، هتخليني اطلع نتيجه احسن، وممكن اغير من انواع السبيشيز بتاعت الحيوانات. So in order to understand that, we need to be familiar with the comparative toxicogenomics. And actually, it is the interaction between 3 major players. The drug, which can affect the disease, lead to the disease, it can affect the genes, and of course, there is a close relationship between the genes and disease. There is drug-gene interaction, there are drug-disease relationship, and there is gene-disease relationship. If we understood these 3 interactions or relationship between these 3 players, we can understand and generate a comparative toxicogenomics. Keep in mind that these science, the toxicogenomics, actually come from different sources. The raw material, the inputs of this branch of science, from data from published literature or articles or studies published, data from studies in the lab, data from traditional tests like histology, blood analysis, urine analysis from human. All of them need to be combined, evaluated, and interpreted, and do statistical analysis by bioinformatics to give a full picture, which is called signature for any drug. فهمتوا اللعبة يا شباب ولا لا؟
Yes, doctor.
Okay.
Yes, doctor.
So let's summarize what is toxicogenomics, and this will be the last slide of our lecture today. What is toxicogenomics? So we have an animal now. This will do some traditional tests. So this animal have been treated with a certain drug. This will be do some analysis, which is the traditional analysis, like the case of histopathology, to check if this drug have affected a certain organ. So normally, toxicologists will do a lot of histopathology for all the organ of that animal. We can do clinical chemistry, the blood, test the blood for any variation due to the treatment. Then we'll do some weight and physiology tests as well for the animal. And then we'll measure the absorption, distribution, metabolism, excretion of that drug in that animal. And of course, all of these methods are called the traditional methods. This will be pooled into a toxicological database. So we now have these all results came from one drug, second drug, third drug, different tests, different responses. We collect all this information for a lot of drugs. وعشان تفهموا الموضوع اللي احنا بنقول عليه، الكلام ده ممكن ما يتعملش عملي، ان ممكن انت باستخدام البايو انفورماتكس تجيب كل المعلومات دي من الاستاديز اللي معموله اصلا اونلاين، تجمعها بدل ما تعمل كل التجارب تاني. لا، انت بتاخد نتائج اوريدي اتنشرت في الابحاث العلميه، ان انت بس مجرد بتجمع كل النتائج دي عشان تعمل بيها الداتابيز جايه يا اما من شغل عملي اتنشر يا اما من شغلك. فهمتوا الفكره لغايه دلوقتي ولا لسه؟
Yes.
All this information from the traditional techniques will be pooled into a toxicology database. نيجي بعد كده. خلاص. At the same time, you can get from the literature or even from experimental procedures, you can do gene, protein, metabolite expression profiles. So احنا عندنا كده ممكن الكلام ده كله يتحول ان انا اقدر اجيب الانيمال اللي هو untreated واجيب الانيمال اللي هو treated واعمل جين، بروتين، ميتابولايت اكسبريشن بروفايل، وابدا اشوف الفرق نتيجه للتريتمنت ده. هل اغير مستوى الجين عمل ميوتيشنز، هل اغير على مستوى البروتين، هل اغير على مستوى الميتابولايت او الباي برودكت بتاع البايوكيميكال رياكشنز، وابدا اسجل الكلام ده برضو على الكمبيوتر. خلاص اهو اروح جاي مدخله في حاجه اسمها اوميكس. ولازم تبقى فاهم ان كلمه اوميكس يعني ايه. اوميكس بتتربط باللي بيتعمل. لو انا شغال على الجين يبقى اسمها جينوميكس او جينوم داتابيز. لو انا شغال على بروتين يبقى اسمه بروتيوميكس او بروتين داتابيز. فهمتوا قصه كلمه اوميكس ولا لا؟ اوميكس يعني معناها خدت داتا حطيتها على كمبيوتر داتابيز. فهمتوا؟
Yes, doctor.
Okay, so how many database we have now up till now?
2.
2 toxicology database and genome database، صح كده؟ وكلهم لهم علاقه بنفس بالدراجز اللي اتعملت، صح؟ طبعا
Yes.
compared with the untreated عشان اقدر اعرف الفرق. فاهميني؟ اوكي. بعمل حاجه اسمها sequence anchoring of molecular expression. يعني ايه sequence anchoring؟ يعني بمنتهى البساطه ابدا اشوف السيكونس اللي حصل له افكت نتيجه للدراج ده بيديني حاجه اسمها ده عن طريق ايه بقى الكلام ده بيتعمل سيكونسنج انكرينج. بربط كل سيكونس اتغير في هذا الجينوم بعلاقته بالدواء. هل هو فعلا مرتبط؟ هل هو فعلا reported in different studies that these drug or these toxicant will affect the gene sequence, because we are talking about genome database here, for example. So this actually is done by computational analysis. Now we have a multi-domain which contain a multi-gene. And if we're talking about multi-genome, that means we can have different genomes. And this actually a knowledge base database. Of course, you need a literature mining. يعني ايه literature mining? You need to search in the literature or the published studies to add to this knowledgeable database all other info that exists in the studies that is published. So we have responses from these drugs that have been examined by traditional methods. And then we have all computerized data came from the sequencing, and we have done some sequencing anchoring, and we linked the drug which affect a certain sequence in the gene. So we need to do something called phenotypic anchoring. We need to do with the computer of the bioinformatic, trying to align the response that came here into the toxicology database with the response, with the correlation that have been seen due to the sequence anchoring. So in other words, you will identify the gene or protein or the function groups or the pathway or the network of pathway that is correlated to a certain response here in the mouse or in the animal. This after phenotypic anchoring, you can do biological modeling. So you do biological modeling, try to link both together, and this will lead to, if you do this for one system, you can do to a different system. You can do for, if you are talking about the liver, for example, you can do for the liver of different individual, the liver, the kidney. So by the end, you have integrated systems toxicology that will interpret the effect of drug on different systems of the organisms at different concentration all over a broad range of input, either from the different database. Okay. في حد عنده سؤال لغايه دلوقتي؟ Any question يا شباب؟
Clear, doctor.
يعني hopefully انتم مركزين معايا، لان انا يعني ما ببقاش مستريح لما الناس تفضل تقول clear. فاهمين قصدي؟ anyway يعني الحاجه واضحه ولا انتوا عايزين تخلصوا المحاضره وخلاص؟
لا clear.
طيب هنشوف. Okay. So this concludes our lecture for today. Okay. And since everything is clear, so hopefully seeing you next week. Okay.
Doctor, thank you.
Sorry.